So for this experiment, biochemical testing for microbiology, I have set up my my desk for my experiment. So first things is we wash our hands, and then we read. Oh, sorry. First thing is we read all the instructions to make sure we gather all the supplies needed for an experiment, so it's all organized. And then we wash our hands and collect all the supplies. I do have my gloves, which is first protection for infection control. It does ask for goggles and apron. Like I said, there's no splashing involved in this experiment, so I'm not wearing any goggles. And I have an inoculation loop that we will use to gather culture from our specimen from our culture. We have the pipettes that is needed to draw specimen from our broth, which is here. This is for the metal red experiment, and we would need that to put 4 drops of that to our MRVP broth, which we'll be using for the metal red experiment. We have our cup with the alcohol to sterilize our inoculation loops, and also our pipettes. I put extra cup here so I can put 30 pipettes and 30 inoculation loops in there that I have to sterilize after the experiment. I have the alcohol here. I will need 4 test tubes for the catalase testing, and which is part 3 of this experiment. And I have the marker, which we have to write E. coli on 2 test tubes, and the Staphylococcus epidermidis on the other 2 test tubes. And then we have the hydrogen peroxide needed for the catalase testing. And I have saved here a sterile little cup that I will pour hydrogen peroxide, so I can use a little bit of it and not cross-contaminate the whole bottle. So for the first part, we will need to culture our specimen. So it asks us to grab a specimen from our nutrient broth to the MRVP broth to culture our specimen. And like always, it asks us to disinfect the tubes, the opening of the tubes, by using candle and heating up that lid, the open lid. I forgot to mention, these are the metal red and the voice testing that we'll be using. I grab a scissor so we can open it right here. So after we gather and transfer some of our culture or our specimen to the MRVP broth, we will incubate it for at least 48 hours, so there will be growth. And after that, we moved into putting the metal the testing of the metal red. So on the second part, we will use the broth from the new incubated specimen, where remember we have to put the names. So E. coli on the E. coli test tubes, and epidermiditis on the 2 test tubes, we have to make sure we put equal amount of the broth in the in the test tube. So the first 2 test tube, we will drop methyl red, 7 drops each, and we can see the reaction right away. It's either gonna turn yellow or no reaction, or it will take a pink tint, like a color, like a reddish color, which means positive test. On the third part of the experiment is the catalase testing, which will be using the hydrogen oxide. Will kind of show the aerobic metabolism because of the oxygen or the bubbles that it will show up. So for that experiment, we would need the culture from our agar plate using the inoculation loop. We have to sterilize the inoculation loop by dipping the inoculation loop in the alcohol, letting it dry, and then taking a culture from our plates, and then we of course will use our marker again to mark our slides, our blank slides, and it's best to mark a round area in the middle so we'll know where to put our specimen, and also mark where it is because it's usually just clear. And then we put a little drops of hydrogen peroxide and see how that will react. Bubbles means it's a positive test, no bubbles means a negative test, and that's how I'm gonna be doing my laboratory experiment. And it tells me also to make sure I sterilize this the test tubes and these slides by pouring alcohol or submerging them in the alcohol and let it air dry so I don't have cross-contamination. And then after all the tests, we can put everything on the bleach solution and for like at least 10 minutes, and then throw anything that is disposable. The one thing that I think that needs to be done also that I forgot to mention during the Voges test is that you actually need to wait 30 minutes for a reaction to show up. That you'll see red, the red coloring on top of the solution. That will mean a positive test. So that is how you're gonna do this experiment. So I'm gonna head out and do mine. Thank you.